Hello, everyone. Welcome to the Cancer Center Symposium. I literally just came from the airport, so I didn't get a time to confer with Jay here, so I'm going to do this completely by memory. And he was recruited a few years ago. So Jay Hesselberth did his graduate work at my undergraduate alma mater, University of Texas, where he cut his teeth on RNA biochemistry. And then he went on to do something very different at the University of Washington in Seattle, where he really got into genomics and development of new technologies. And he's really taken that love of both RNA and genomics and understanding mutational processes and changes in RNAs and how we can detect them to really develop some fantastic new technologies. So not only is he developing new technologies, but these technologies are allowing his lab to basically get insight into some really basic biochemical processes that we really didn't have the ability to get such insight before. So Jay is an associate professor in the Department of Biochemistry and Molecular Genetics and a great colleague. So welcome, Jay.
Okay. Thanks, James, for the introduction. So today I'm going to tell you about a new method that we've developed in the lab over the past year or so, I'd say, that involves looking at DNA repair capacity in cells. And I'll talk about that for about half the talk. But what the method has allowed us to do, I think, is start thinking a little more broadly, much more broadly than the assay itself, about new methods that allow us to measure what I'll call functional heterogeneity in populations of single cells. And I'll talk about that at the end. So the method that I'll describe was spearheaded mainly by two people in the lab. Mandy is a graduate student who just passed her comprehensive exam. So she can get back in the lab and start doing experiments again. And Shannon Walsh is a postdoc in the lab who has been kind of developing methods that are riff off what Mandy started with. And then I'll show a little bit of data from a medical student that was in the lab this summer, Lakota Hardy. Okay, so to start out, just make sure we're on the same page. So DNA is this amazing molecule in cells. We all know the chemical diversity of DNA is quite broad, and its chemical diversity is both, what makes it fantastic during normal cellular processes, but also is its Achilles heel. Because the bases, the sugars, and sometimes the phosphates can be modified by either just basic metabolic or cellular processes, but also environmental exposures. There's ways that cells have developed to recognize what I'll just call broadly DNA damage and then repair it, often ideally back to the original base, but sometimes not. So here I'm just showing you, a few examples of DNA damage. So mismatches can be created during DNA replication. Ribonucleotides can be incorporated during DNA replication. There's specific pathways that pull ribonucleotides back out of DNA and replace it with a deoxyribonucleotide. Uracil in DNA can be incorporated two ways. One is direct incorporation of DUTP, and the other is deamination of C. And then there's a bunch of types of DNA damage that are created by environmental exposures. So, for example, alkylation and oxidation. Oxidation when you have molecular oxygen around, you get 8-oxy-G. Pyrimidine dimers caused by UV light, and then platinum adducts and alkylations caused by, for example, chemotherapies we use. And we know that the repair path, so there's a bunch of repair pathways that the job of these single proteins or complexes is to scan DNA for problems that they recognize and then ideally convert it back to the original base. So mismatch repair takes care of mismatches. Two things can recognize ribonucleotides in DNA, RNase H2 and topoisomerase 1. Base excision repair encompasses a whole class of enzymes that recognize single bases usually and pull those out. And nucleotide excision repair is a repair pathway that recognizes bulky adducts and repairs those back to normal. So one thing to note here is that all of these, except for direct reversal, all these other pathways initiate with strand incision. And that's one of the signals that we follow in the assay. So that's kind of a key idea that you'll see. Direct reversal, by contrast, that pathway catalyzed by mainly two proteins, MGMT and ALKBH2, and those proteins are essentially, they're made for a single use. They are suicide proteins, so they recognize alkylated G, for example, pop off a methyl group, and then that protein's dead. It's a suicide reaction. You have to make a new protein to repair it, to do a second round of repair. So direct reversal is a case where the DNA strand is not incised. And we know all these repair pathways are important because if you have mutations in many of the genes that encode these proteins, you end up with a number of diseases. So, for example, mismatch repair leads to colon cancer, and then mutations in nucleotide excision repair factors lead to xeroderma pigmentosum, which is kind of a predisposition for a number of cancers, and then a variety of other different types of cancer. So we know the repair pathways are important for kind of normal functioning, and if you mutate them, cells gain the ability, for example, to grow uncontrollably sometimes. And the repair pathways are also involved in, so once you have the disease, the repair pathways are also involved in responding to many types of chemotherapy we use. So for example, cisplatin creates platinum adducts on often GG dinucleotides to cause problems during DNA replication. And then the drug temozolomide is used often in glioblastoma that creates methylated guanines, and those are often repaired by either basic excision repair or direct reversal. So not only are the pathways are important for normal processes, but they're also important for response to chemotherapy. So, we got interested in thinking about how to measure the ability of cells to repair DNA. And, basically we'd actually developed some other methods that I've talked about in the symposium previously. And to make a long story short, those methods worked and they're great, but one of the guys that won the Nobel for discovering nucleotide excision repair started scooping us and doing the exact experiments we wanted to do. So I thought maybe it was time to pivot. So I started reading the literature and came across kind of a body of literature that discusses this idea of measuring DNA repair capacity in cells. And so the idea here is that every cell has some intrinsic ability to repair its DNA. And that ability can vary by genetic background. So, for example, just as I showed you before, these can be mutations in BRCA genes, for example, or P53. And it can also vary by environment, where if you have too much sun exposure and you're Caucasian, you tend to develop skin cancer faster than others. Or if you're exposed to cigarette smoke, you develop lung cancer. So one useful way to think about DNA repair capacity is as a trait, as a quantitative trait. So meaning everyone's genetics is projected on DNA repair capacity, and I would bet that everybody's cells in the room has you have a slightly different intrinsic repair capacity, and that might predispose you to disease, but also might change the way you respond to chemotherapy if you were given some, for example. And we know that DNA repair capacity can have a big effect on therapeutic efficacy. For example, people with BRCA mutations, if you have a BRCA mutation, you're actually sensitized to therapy with PARP inhibitor, meaning repair of double-strand breaks in the absence of BRCA relies completely on PARP. So now if you inhibit PARP, cells tend to apoptose. So we know there's quite a bit of a genetic component to DNA repair capacity. Okay, so there's many ways to measure this trait, and I'll just talk about some of the ways that you probably know and why we don't think it's probably the way forward. So this is one of the oldest, the COMET assay, and the idea here is that you want to measure damage at the single cell level. So what you do is you embed cells in agarose, and then you stain their DNA. In this case, it's green. And if you do that for basically any cell, it'll appear as a sphere. So now if you apply an electrophoretic field, the DNA tends to migrate out of those cells, and that's dependent on damage. So the more double-strand breaks you have in that DNA, the more you'll tend to create this tail, and that structure gives the name comet. So the longer the tail, the more damage is the idea. And people have used this and actually, tried to, for example, quantify the tail length as an indicator of the amount of damage. I've run this assay. I personally feel like this is the kind of assay you can get any answer you want out of the comet assay, depending on how you run the experiment. It's not a really robust assay. And the bigger problem, from my perspective, is that you don't learn much about the particular types of damage that are happening in those cells. There's also damage microarrays. Some of these are kind of a prelude to the method I'll talk about. So, for example, Sylvie Savago has developed, using microarray technology, a way to monitor DNA repair in extracts where she prints double-stranded DNA substrates that have particular lesions and have a fluorophore. So you print a microarray full of those oligos, and then you incubate that microarray with cell extract, and DNA repair activities in the extract act on the substrates, and you release the fluorophore, and then you just simply scan the array after the extract and try to ask, how much repair activity was associated with a spot where, for example, you put down a permanent dimer, and now the permanent dimer got cut in the extract. Again, useful to a degree, and she's shown that you can apply this method, for example, across different cell lines and show different levels of repair activity, but it doesn't give you a whole lot of information at the level of the repair events themselves, and it's certainly not scalable to single cells. A third one is the host cell reactivation assay. So this is developed by Leona Sampson at MIT. So what she's shown is that if you can create plasmid reporters that have a fluorescent protein, so GFP, RFP, YFP, and then embed in those genes, the genes for those fluorescent proteins, a particular type of adduct, so a permanent dimer, ADOXO-G, or Atheno-A. And then if you transfect these plasmids into cells, only after repair happens will that gene be able to be transcribed normally and lead to green fluorescence, red fluorescence, or yellow fluorescence. And so this assay is, by its nature, single cell. In other words, it's flow cytometry. You're measuring fluorescence of single cells. But it's limited by, first of all, you have to make a particular plasmid, which is somewhat laborious per repair substrate, and then you're also limited by transfection. So some cells will get one plasmid, some cells will get three, and by its nature, you have to have cells that are transfectable to be able to run this assay. The same goes for an assay that Larry Loeb developed a couple of years ago, and this is what kind of sparked my interest in developing a new method. Is this thing called, he just called it the oligonucleotide retrieval assay. So it's a very simple assay. You create a DNA hairpin that has a permanent dimer and has a biotin. And you take that piece of DNA and you transfect it into cells, and then you wait a day, for example, and then you blow the cells up and do a streptavidin pull down, and then just assay repair using qPCR primers on either side of the dimer. So the dimer will inhibit PCR, polymerase can't go through the dimer, and the more repair you get, the more qPCR signal you get out. So this is a nice idea, but again, it wasn't at the time amenable to single cells, and you have to have cells that you can transfect. So those are kind of the big problems we were working with. Although he actually developed a number of assays that he never published that are in a patent that never got filed. So I think he was onto something, but maybe was just a little bit too early with the thinking. So we've actually kind of combined a number of these methods to develop a new way to look at repair capacity in cells. And so essentially what we do is what the Loeb lab developed, so this oligonucleotide retrieval assay, except we change the machinery around a little bit. So the idea here is that we put the substrates on beads. We put them on beads for reasons that will become clear later, but cut to the chase, it's to be able to do analysis in single cells. And on those beads, we have oligonucleotide substrates that have this little red bit is a stretch of DNA that has that's a unique sequence for a particular bead. And then coded on each other of those sequences, oligonucleotides, we've attached DNA repair substrate as a form of a hairpin, and the colors represent basically different types of substrates. So uracil, ribonucleotides, permanent dimers, oxidations, methylations, so on and so forth. So we make a preparation of these beads. We mix the beads with cell extracts. Currently, we've done bulk, but as I said, we're moving to single cell. And then what happens, as I said, most of the repair activities initiate with strand incision. So if repair factor A is in the mix, he's going to recognize the substrate, the green substrates, and create some nick on that substrate and leave an end. And so the key thing in the method is that under these conditions, we're basically following creation of new ends in the assay. And as you may have seen me talk, we named the method. So we're measuring strand incision on DNA hairpins. So we named the method haircut. Okay. Tough crowd again. I always get the just blank stares, but anyway. Okay, so the way the method works is as follows. So again, as I described, we have beads, we synthesize or attach. Currently, you can just directly attach some of these oligos to beads, but for single cell, you have to directly synthesize these on beads. But the upshot is that you'll have an oligonucleotide that has a bead ID and a unique molecular identifier. So the bead ID, when you collect that by sequencing, will associate particular repair events to a given bead. And the molecular identifier is essentially a counter. So this is a fixed sequence per bead, and this is a random region. So many oligonucleotides will have different UMIs. And then we mix those beads with a substrate that has some query position. So again, that's, for example, a uracil, and a substrate ID. So this is another sequence that's unique to the class of repair. And then we just simply attach those together by splint ligation. So we just design it up so that we can anneal a splint. These two oligos create a nick. We add ligase, they get attached. So the reason we did this was so that we could make a mixture. So in other words, if we create a bunch of different types of DNA repair substrates that all have constant sequences out here, we can just simply mix them together and create beads that are now coated with a bunch of different colors. Repair substrates, right? So every hairpin on this bead has a common bead ID, that's the red bit. They have counters, that's the colored bit, and then a different type of repair substrate. Okay, so this is how we read out the signals in the assay. So we take beads, mix those beads with different types of extracts. So again, this is bulk extract at this point. And what happens, and again, if there's a repair activity that recognizes the particular green substrate, it'll create usually a nick or a gap. So that's the key event. So now we wash the beads away. We wash away sort of non-covalently bound DNA. We end up with free 5' ends, and then those are recovered in a fairly standard sequencing library preparation where we lay a primer down. The 3' ends fixed on the bead, so we lay a primer down, do second strand, ligate double-stranded adapters, and PCR. So when we sequence, we come back at the repair product from this direction so we can see exactly where repair happened because we're sequencing the products of repair directly. So we're capturing the incision products. Okay, so I'm going to just walk you through a couple of examples of what the data look like. So this is for a substrate where we've programmed the hairpin with an AAU base pair. And A will be at position 11 and the U's at 36 in a plot that I haven't showed you yet. But if you put this piece of DNA into extract, the uracil DNA glycosylase will scan this bit of DNA until it sees the uracil. It'll pop the base out and then clip the glycosidic bond, leaving an abasic site. That's what that 0 with a slash through it means. So you've still got phosphodiesters on both sides of that guy. He just doesn't have a base. And there's a different protein called the apurenic endonuclease that recognizes the abasic site and cuts both phosphodiesters, leaving a single base gap. So what you should see, if repair is going on in the extract, remember the bead here is attached to the 3' end. When we run primers back and create libraries, we should see capture of position 37 in the plot. So now I'm just showing you, this is about 50,000 reads from a high-throughput sequencing experiment, and I'm just mapping the 5' end of each of those reads back to the sequence of the hairpin, and then I'm just calculating the density of those reads. So roughly 80% of the reads mapped to this position in the green case. So we've done this experiment for a couple different lines. One is just a wild-type haploid human cell line, HAP1. We have an UNG knockout in blue, and then we also, at the time, we're using HCT116. So what you can see right off is that when you have wild-type or HCT116, which you expect to be normal uracil repair, you can see that the predominant signals are right at position 37, which is where we would have predicted. And if you knock out UNG, that signal goes down. It's not gone, but it's reduced by about 80%, and there are actually backup mechanisms to repair uracils in those cells. So what this told us was that, first of all, the assay worked, kind of, right? We could program the system with a substrate, recover the signal we expected, and then a second thing was that if we modulate a DNA, if we delete a DNA repair factor, in this case, we can see a change in the assay. Okay, so I'm just going to show you a few more examples. We've programmed the system with lots of different ribonucleotides and have seen a bunch of different things. This is actually an old result, so this is one of the first times we did this experiment. So here's instead of an AU pair, it's a GU pairing. So GUs are recognized in principle by two different pathways in cells. One is standard base excision, so that's the pathway I showed you before, and you should create a single base gap. And then it's also seen in cells by mismatch repair machinery. So this was a prediction, and so again in the signals what we can see is that for the base excision repair at position 37, we can see that robustly. That's that big signal at position 37. Again, it goes down when you delete UNG. But then we also see signals 8 base pairs upstream and 8 base pairs downstream of that uracil. We thought potentially that was mismatch repair, but it actually doesn't change all that much in HCT116 cells, which are kind of defective for mismatch repair. But it does change when you delete UNG. So in other words, for the signals 8 base pairs up and downstream, the signal goes down at position 37. You lose the upstream cleavage in the UNG deletion, but you seem to gain the signal downstream. So we don't understand what this activity is. We think it's mismatch repair, but this is one of the early results in the assay. And then I'll just show you a final example of what happens if we program the system with a ribonucleotide. So here we put a ribo G across from C on the substrate. So ribonucleotides are repaired in two ways by cells. One is ribonuclease H2 recognizes ribo G in DNA and clips the 5' position. So in this case, again, if we capture the product, or really the intermediate of repair, we can see that the 5' end should be at position 36. And then topoisomerase 1, actually fairly recently, Tom Kunkel and Peter Bergers worked out a pathway by which, and Sue Jenks Robertson was involved too, a pathway by which topoisomerase 1 will cut the downstream base of ribo G, the downstream position of ribo G, and also cut a few bases upstream. So now if we look at the data we get, so here, this is position 36. So this captured product, we believe, is RNase H2. We haven't shown this by a deletion, for example, but it's at least consistent with what the prediction is. And then the other sites, again, are 1 base downstream and a few bases upstream, we believe might be topoisomerase 1. So as soon as I say topoisomerase, you should be thinking drugs, because you can drug some of these enzymes. And I'll tell you, we've tried the experiment where we supplement the extract with a drug that inhibits topoisomerase 1 called camptothecin. So the idea was that when you add camptothecin to the extract, you should reduce topoisomerase 1 cleavage here and here. And it didn't work, meaning it didn't really change plus and minus camptothecin, but we're still in the middle of trying those experiments. And so camptothecin actually stalls the cleavage complex of topoisomerase 1. So the idea was actually that there'd be enough DNA in the extract that topo 1 would get stuck on the DNA from the cells and be unable to cleave the hairpins we program in the assay. Okay, so we've done this for a bunch of different types of substrates, ribonucleotides, mismatch uracil. Basically, I think we've done everything on this list. We haven't done platinum adducts yet, but that's coming. One interesting one that we haven't done yet, but you can put in synthetically, are the therapeutic nucleotides themselves. So you can put in 5-fluorouracil into the hairpins. You can put in zebularine into the hairpins. You can put in Ara-C into the hairpins, and then watch what happens at the level of repair. So one of the big pushes right now is to get as many of these working at the bulk level so that we can interpret signals when we move to single cells. So that's basically where we were at, I don't know, a couple months ago, where I gave a short talk, and I just wanted to show you a few other pieces of data that we think are going to improve the assay. So one is data from Lakota, who was a medical student that was in the lab for the summer. So most of the, if you read sort of old literature on using extracts for DNA repair, almost universally they incorporate ATP regeneration systems into the extracts. And we weren't doing that initially. We were just making extracts and adding in the DNA. There's a good reason to do that, to supplement the extracts with ATP, is because once you make the extract, the machines in the extract are hydrolyzing ATP all the time, and that gets depleted. But in some cases, the enzymes that the extract would be using to recognize DNA and unwind the helix require ATP. So this is just an example of how, if we're looking at nucleotide excision repair, there's a couple of enzymes that actually use ATP in the repair pathway. Once the bulky adduct is recognized, the strands have to be unwound to expose a single strand that's all ATP dependent. So now if we make extracts and look at, this is cleavage of a thymidine dimer. If we look in extracts with HAP1, we expect, again, in this case, the hairpins immobilized down here, the beads attached to the 3' end of this little bit. So we expect a signal sort of on the 3' end of where the adduct is. So the dashed line here is the adduct, and most of our signal is a few bases downstream of the adduct, actually where we expect it. And I'm not showing you this, but that is modulated. We've in a deletion of XPG, I believe. So in an XPG mutant, this signal goes down. In this case, I'm just showing you that if you add an ATP regeneration signal, signals go up in this assay. And it was true that we weren't just looking at nucleotide excision repair under these, with these beads. We were looking at a bunch of different types of repair. So ATP regeneration systems generally improved our ability to recover signals. So that's one thing that we should have thought about from the get-go, but we'll probably be including going forward. And then we can also play lots of different types of tricks by supplementing the extracts. So this is an experiment. So let me back up. So one of the issues is the assay is really good at seeing the intermediates of repair, meaning strand incision, you nick the strand, and then we capture that nick, or we capture a gap that's made. But if the extract continues on and actually synthesizes new DNA, ligates it back together, those are invisible. Because we've lost the intermediate, now you're at the product. So we really can't tell the difference, we don't think at this point, between the substrates and products. So we've been relying on intermediates. But we can play tricks to try to capture different types of intermediates you'd expect to accumulate. So this one is actually, the substrate we're programming the system with has an abasic site initially. So just, as I said, repair of AU goes through an abasic site intermediate. Now we're just putting it in directly into the extract, and you can see what happens. And these are actually repaired in two ways. So one is, as I described before, so-called short patch repair, where APE1 will create a gap. And at that point, you've got a nick in the strand, and the first base is a deoxyribose phosphate, so it has no base. Pol beta synthesizes one nucleotide and then also clips off that DRP and ligates it back together. So in short patch repair, you should see most of the signal accumulate one base downstream, just as I showed you for the repair of the AU substrates. And the other way this substrate can be repaired is through long patch. So in other words, APE1 initiates the reaction by cutting the 5' position of the A basic site. Pol beta synthesizes usually one base, maybe two, but then another DNA polymerase takes over, makes the blue bit, and I haven't drawn the intermediate. That would create a flap that's then cut off by Fen1, and then you ligate the strands back together. That's kind of full repair. So now the question is, what happens? So in the red, this is just standard extract, so we can see short patch repair, but we also get signals downstream that we believe are long patch events, meaning what's happened is that you've extended and the flap's been cut at a new position, and we capture that position down here. But now if we add dideoxynucleotides, this is dideoxy-TTP, we should get stalls at very specific sites where there's an A in the template. And as it turns out, there's an A in the template it's right here. It's +2 downstream from the A basic site. So when we add dideoxy, we can see a little blip in the signal. And again, what we think is happening there is that we've stalled a long patch repair event at that particular site. Now we can capture it in the assay. So it looks like there's actually more complicated repair happening in the assay that we didn't expect, and that we'll probably have to deal with in the future. But some of this I think we can get at by the right type of supplementation strategy. Okay, so that's kind of where we're at with haircut. And so I just wanted to talk a few minutes about kind of why we started to develop this assay. And really the motivation was to have a new way to assay what I'll call functional heterogeneity. So there's a bunch of new methods now where we can measure at the level of single cells sort of the levels of different biopolymers in the cell. And the most common one at this point is mRNA. So we have one of these machines on campus. You can look at mRNA expression profiles in single cells. It's very robust. As long as you're not an immunologist, it's very robust. Turns out immune cells are a little bit refractory to the method, but hopefully they work that out. So there's classes of assays that just measure levels of mRNAs. And some of the more clever ones that are coming out can actually measure levels of, for example, mRNAs and proteins. So there's this nice method from Rahul Satij's lab where they're actually measuring both mRNA and cellular epitopes, proteins, simultaneously on single cells. So this is kind of a clever trick where the basic idea is shown here. So again, for single-cell RNA-seq, you're going to encapsulate cells and beads with particular functionalities on them. So if you're capturing mRNAs, usually the beads have oligo-DT. And if the machine we have here is from 10x Genomics, so it's a droplet maker. So what it does is you've got a dilute solution of cells, a dilute solution of beads that get pinched off, and then you're just creating drops at some high rate, a couple thousand per second usually. So many of the drops don't have anything in them. Some of the drops have one bead, and then every once in a while you'll make a drop with a cell and a bead. And the beads are in lysis buffers, so as soon as this drop's made, the cell blows up and spills its guts out. And then again, normally you'd capture the mRNAs via the bead. So what they did here was a clever trick where they basically wanted to measure cell surface epitopes using the same assay. So what they did is they create antibodies that recognize cell surface epitopes, CD4 for example, and on the antibody they put a synthetic poly-A tail with a tag they can follow during sequencing. So what happens is they make a mix of cells with the antibodies, they wash away unbound antibodies, and now each of these cells is coded with some distribution of antibodies that each have a unique tag that can be read out using the standard oligo-DT capture. So when this goes through the assay, you collect the sequences, you're reading out endogenous mRNAs and the synthetic tags that got put on the antibodies. And now here what they can show is that, so this is a standard t-SNE plot where every dot in the plot is a particular cell, and the different axes represent the variation in this case, mRNA expression per cell. So you can get, so what's shown here is sort of a cluster of B cells that have a B cell signature, a cluster of T cells that have a T cell signature. Okay, this is sort of standard single cell RNA-seq. But now down here, they can also look at the epitope tag. So here they're showing what happens for CD4. So here they're showing where CD4 mRNA is captured. And you can see only a few cells have CD4 mRNA, and they tend to be T cells and monocytes. But now with the antibody tag, the whole cluster lights up. So those are CD4 positive T cells. So this method is a nice way to kind of integrate both mRNA and protein levels, and you're just limited by the antibodies you can make in this case. So what developing the haircut methods got us thinking about is assaying functional heterogeneity. So instead of looking at levels of mRNA or levels of protein, we want to directly measure enzyme activities in single cells. So, and this could be any, if you think about any substrate intermediate product relationship, I think we could probably measure it in single cells. So again, the idea here is just like the 10x platform, we now have a droplet maker in the lab from Dolomite Microfluidics. We have the ability to make haircut beads. And so the next experiments basically involve doing haircut, but at the single cell level, where you're making drops every once in a while. A cell goes into a drop with a bead. The cell lyses, spills out repair activities, and then acts on the activities act on the bead, and we just recover signals like we do normally. So basically, the enzyme activities are limited spatially by the droplet, but then once that's done, we stop the reaction by breaking the emulsion, and then prepare libraries in bulk. So the goal here would be to, for example, quantify repair of, let's say, 100 repair classes, AAU, GU, oxidations, methylations, bulky adducts, so on and so forth, in each of roughly 10 to 100,000 cells. And like any single cell RNA experiment, you become sequence limited. So I think we can easily get enough information to quantify 100 substrates in 10,000 cells, but actually doing 100,000 cells may be cost prohibitive. But that would be kind of a bigger goal. And then, of course, we'd like to apply this method to measure heterogeneity in complex mixtures, to measure DNA repair capacity in single cells. And this also, I think, will enable all kinds of other experiments to do genetic screens for things that modulate DNA repair. Okay, so what would you expect from this kind of assay? So there's a couple of things to think about. So imagine you've got a population of cells. And I'm just going to, actually Mandy drew this figure. I think it's a great figure. So you can draw it up a couple of different ways, thinking about what types of things you might see in a population, and then what outcome, what the outcome might be if these were cells from a patient undergoing therapy. So for example, if you run the assay and you're measuring repair capacity, and here I'm just using repair capacity as just a general phenotype, not a specific type of repair. So one outcome might be you run the assay and you get some normal distribution, meaning that all the cells have roughly similar repair capacities. So the interpretation here would be if you add a drug that kills these cells, you'd probably get a good response, and the recurrence would not be likely because they're all going to respond in the same way. You could also have high variance, meaning you get a bigger spread in repair capacity, and obviously we don't know what these two things look like until we're able to compare them. But if there were differences in kind of a narrow distribution of capacity and a broader distribution of capacity here, you might have a response and you might recur simply because there's a bigger variation. You could have outliers, and this is known to happen, where you measure repair capacity and there's a normal distribution, but every once in a while you get single cells that don't look like the others. Their response is response, broadly defined as likely, meaning you can see a bulk population disappear, but then you've failed to kill the rare guys that come back. And then a fourth possibility is that you have different subpopulations that are all abundant. And at the level of repair capacity, this would kind of appear bimodal. And here, the response would be unlikely and also likely to recur. So that's kind of just thinking about the types of things you might see in the assay. One of the problems, though, is that, so I wrote this up as a grant, as one does, and got reviews back. And the reviews, there were two main complaints. One was we hadn't discussed quantitation of the assay sufficiently, and I could stomach that. I agreed with it. The other was that they didn't like the lysis buffer we used. And to make that complaint, you had to ignore a whole bunch of data that said the lysis buffer we use is just fine. But anyway, I digress. So we've been thinking about how to really quantify signals in the assay, and this is actually not easy. Because if you think so again, we're measuring activities. It's functional heterogeneity. We want to measure enzyme activities, ideally at the level of single cells. And classical enzymologists, if you're assaying an activity, you can vary two things: time and concentration. And so to get activity of an enzyme, to make this double reciprocal plot, each little dot, right, on a Lineweaver-Burke plot is a velocity you've measured of an enzyme under different starting substrate concentrations. And so now, if you plot that line, you can extract all the relevant parameters for the enzyme, the Michaelis constant, and the velocity, and also the slope gives you the ratio of those two things. So here, again, you need both things. You need to be able to vary time. You need to be able to run the assay over a time course to get a velocity, and you need to be able to measure you know the concentration of, in this case, substrates in the reaction. So we don't have time. In other words, the time in the single cell experiment is the time between, is a single time point. It's the time between a droplet is formed to when it's lysed. So it's basically all one time point. We don't really have the ability to run a time course. So the idea is we'll vary the only other thing we can, and that's substrate concentration. So now instead of putting substrates into the system as we have been at sort of equimolar concentrations, now for each, we actually make the system more complicated, but the goal is to be able to measure activity for each particular substrate. Now we have sort of 5 different ones that we can see uniquely by sequencing that span some concentration range. So now, in that experiment, again, we haven't done this yet, but the idea is that under those conditions now, under a fixed time point, you can broadly classify activities into different classes. So over a range of concentration, they may not all be low. That's no activity. Under high concentration, you'll see activities under high activity, you'll see activity under all concentrations. And then, again, theoretically, you'll get things in the middle. So that's the kind of the idea we're taking because we don't really have the ability to modulate time in the assay. So these are kind of headier ideas that I've been thinking about, and I'd love to get your feedback on some of these ideas. So again, it comes back to this question of what would you possibly see? So one thing that I think you might see is if you run the assay and you're looking at lots of different activities, one possibility, so now the basically experiment would be, take a cell or population of cells and you treat them with different drugs, 5-fluorouracil, cisplatin, or a PARP inhibitor. You run the assay after you treat to see how the drug established some changes in DNA repair capacity in that population of cells. So I wonder to what extent the phenotype might actually be heterogeneity itself. Meaning some drugs, and of course this might be cell types dependent, there's all kinds of things that could come out of this experiment, but one might be that if you treat cells with 5-fluorouracil, many of the cells develop sort of broader base excision repair capacity because they're upregulating those genes, but they're all doing the same thing. The same thing might happen with cisplatin. They're all changing nucleotide excision repair to do roughly the same thing. But now, under other conditions, and this is, I'm just making a guess here, where you add, for example, a PARP inhibitor that's going to affect more than one pathway, the phenotype might actually be heterogeneity itself. So by treating cells with an inhibitor that inhibits multiple pathways, some cells might do it one way, some cells might do it another way, and you'd see that in the outcome. And obviously, this would be a valuable readout to understand in a tumor how the cells are changing. Okay, I just have a few more slides. So there's a couple of big questions we want to ask with haircut, but I wanted to get to a couple of other examples that, again, we're thinking about how to sort of push the method more broadly. One is putting in different substrates. So Shannon has been working on, instead of hanging naked DNA on beads, hanging nucleosome particles. So the idea here is that, and I have to give a big fat shout out to Alexis and Maggie and Aaron's lab who have been helping us build these. The idea is we take a piece of DNA that's known to preform nucleosomes, incubate that piece of DNA with histone octamer and isolate nucleosome core particles, and then hang that up on beads as the substrate. And obviously, often this would be the substrate that cell extract, that repair enzymes would normally see in cells. And just in initial experiments, just to describe, to see if we can see repair on these particles, we've put uracil on a particular position, and if we put in nucleosome core particles, we can see very robust capture of uracil repair on those particles themselves. And so we think programming the nucleosomes with different types of repair substrates can be valuable, and there's also a literature from Michael Smerdon and others that have shown that repair efficiencies change depending on where within the nucleosome the adduct is. So, for example, if it's on one face or the other, that changes repair capacity. So lots of sort of just standard biochemistry we can address using the method. And then another one that we've been thinking about, we haven't kind of pulled the trigger on this, would be measuring a more complicated activity, that is histone post-translational modifications on tails. So using the same nucleosome substrates, the idea would be to incubate these beads with cell extract that have active histone tail post-translational modification activities, and then read all that out, not by direct sequencing, but by proximity ligation, where you have an antibody that recognizes a post-translational modification that's formed, and then you link that back to a bead ID by proximity ligation. And once you have this molecule, amplify it up in sequence. And so we think, again, this may be a valuable tool to look at tail modification activities. Some of these probably won't work that well. I mean, we're going to start with ones where the activities themselves are already pretty abundant in cells. But that's something we're thinking about for the future. And one thing I've been thinking about, it's a little bit half-baked, but I wonder to what extent something like this would be an alternative to the way people usually read out changes in tail modification, which is how they're laid down on the genome by ChIP-seq, right? That's there's a lot in between changes in tail modification and laying down modifications on the genome. And I think this might fill a nice gap to look sort of directly at the activity itself. Okay, one more. We'd like to move this to double-strand break repair. We haven't done this yet. We haven't done this yet largely because it's slightly more complicated, although I think I've convinced myself that it's not that much more complicated. And it's more complicated because now the substrate of repair is bipartite instead of a single DNA molecule. So the idea here is that you program the beads with bipartite, blunt, or overhang substrates, incubate these beads with cell extract, components of non-homologous end joining associate, and then sometimes create a ligated repair product, and then you just amplify those up by PCR. And the nice thing is because you would use primers on both sides, the unrepaired substrates shouldn't come up. That's the idea. Again, we haven't done this yet. But so I think this should be possible, and if it was, all kinds of cool things are possible, including because it's sequencing, you can look at microheterogeneity at the ligation junction itself, so you can tell the difference between fill-in, chew back before religation. And I'd love a rotation student to come and do this. I think it's a good rotation product. Okay, so what I'll leave you with is we've been thinking a lot about these methods to measure functional heterogeneity. So a lot of people have focused on, in populations of single cells, measuring levels of mRNA protein. To what end? Probably, to use these levels to predict functional states of cells. So in other words, mRNAs go up or down, and then you predict the cells doing some particular activity. What we've been thinking about is ways to skip this and just go straight to some functional state to see what cells are actually doing. Okay, so with that, I'll wrap up. Thanks to Mandy and Shannon again, Lakota, for contributing an insight we should have had, that is adding more ATP to our reactions. Collaborators that provided tissue and cell lines and funding, particularly the Golfers Against Cancer that just funded it. So thanks. Any questions? Yes, sir.
This is one, but now you seem to have a potential solution. So I've been asking you all, could you basically combine it with like a flow cytometry? So if you took peripheral blood, you'll get cell-to-cell heterogeneity, but you won't be able to distinguish T cells from B cells from macrophages and so forth. But if you could combine it with that antibody technique, but I think you ruled it out on Anschutz before.
So you certainly can, and I'm sorry if I ruled it out. It was the wrong logical path. I think you probably could. Again, you have to be able to link binding events back to the bead, and that's just a general problem. So I think that's totally doable. Yeah. Also, we've thought about measuring specific mRNAs that would be diagnostic for particular cell types. That's straightforward too. Yeah.
Along those lines, it's good about using flow cytometry to link position in the cell cycle with kind of now of course got asynchronous.
Yep. Yeah, so the question is about cell cycle, and obviously repair activities change. Yeah, absolutely. Yeah, I mean, you could use flow. Mandy's, I think, like maybe right now, doing synchronization experiments where you put all the cells into G1 using double thymidine, let them go, and then just watch what happens in a time course. Flow, if the single cell works, great, definitely. I don't know how many cells you'd get back in a cytometry experiment. Probably enough, but yeah. 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 Yeah, certainly. Yeah. Repair activity will probably change through the cell cycle for sure.
Jake?
Yes, sir.
So I was going to ask the same question, whether, I guess your approach to identify the particular types of cells would be PCR diagnostic for the RNA. You can't couple it with RNA.
You can, just like Satyja did mRNA and protein, right? All you have to do is just put the two things together, hang two separate types of activity or biopolymer probes on the same bead. Totally doable.
So the other thing is,
I should say, if you have to be a little bit clever about the workup, meaning ideally you capture all those signals in the same bead workup. If you have to do capture this one way and capture this the other way, it's not going to work.
so the other question is, so you're measuring an intermediate most of the repair.
Yes.
So you're not measuring the whole thing. So based on, if you put in your lysis buffer really high concentrations of didioxins,
Mm-hmm.
can't you stop everything and measure one nucleotide away and increase your sensitivity?
Measure one nucleotide away.
So in other words, they won't be able to join and get religated. So
Yes.
you'll make a termination,
Ah.
right?
Yeah.
And so you've captured every cleavage event if you can get high enough didioxins through the
Yeah.
lysis buffer.
Yeah, Mandy and I were just talking about this. So I think she's basically done like one dideoxy experiment with one dideoxy, T. And she put it in at roughly the same concentrations that you expect in the extract. So she hasn't flooded the system. But you might be right.
Then if you're only measuring 5% of the activity, right?
Right.
You're healing it.
Yes. Well, that's the question is,
In the context of a single cell experiment,
yeah.
that would really improve
That would improve sensitivity, absolutely at the level of single cells. So maximizing recovery of short-lived intermediates in the assay is paramount when you move to single cells. I want to be able to see every single event because it's not going to be that efficient at the single cell level. Yeah?
Just continue the position of only having people from the department ask questions. So could you do a sort of derivative of what Dick was referring to, but put that dideoxy, so in your construct, you put 3 bases down, so then you would actually be measuring both cleavage
And long patch?
In other words, you would basically get 2 readouts in the same.
Yeah, you could design, you can play all kinds of tricks with careful substrate design. And there may, and you're right, there may be a way to set it up so that you maximize the ability to capture an intermediate, or like an intermediate of the 2 pathways, for example. Yeah, essentially. I mean, there's nothing specific about DNA. I'd love to measure modifications on RNA and put in a substrate of a small molecule substrate, read it out with an antibody to the product, right? There's nothing, so I think the assay is very scalable to all kinds of functional readouts if you dream it up right. Anyway, okay. Thanks.